All right, thank you. Welcome to our last science luncheon of the semester, the first, I guess, ever scientific showcase for the Center for Vital Longevity. I am happy to introduce for our first presentation today, we're going to have a brief presentation from Dr. Marco Pipoly from the Wig Neuroimaging Lab. Come on up.
Thank you. Hello, everybody. Okay, so today I'm going to talk to you a bit about the work that I've been completing in the lab since I joined last year, the title of which is Age-Related Loss of Functional Specialization Across Multiple Scales of Organization in Human Brain Function. So I think it's going to be good for me to start you guys off with this big picture idea that we had when we approached the project, when we were thinking about what our goals might be to understand this idea of loss of functional specialization within. And so I think it would be great to orient you guys to how we're thinking about the central nervous system in terms of its organization and a hierarchy, where you can imagine as a researcher, you might be interested in understanding brain function, maybe brain function change as a function of the aging process at any level of this hierarchy. So as someone who uses functional magnetic resonance imaging, I often find myself thinking about this at the level of areas and systems in terms of what I have access with my measure of interest. And what we know about areas is that they tend to display some specialized features, right, in terms of the types of information that they process to support larger communication throughout the brain. An example of which can be taken from our nice occipital cortex here, which we know is packed with different areas that are responsible for processing different features from our visual world that are generated and supported out through a system called the visual system of the brain to other areas to support higher order cognition and complex behavior. But what we know from aging research is that as we age, there's an alteration in the selectivity of these specialized areas in terms of how they respond to different stimuli, which we know also affects this system level of organization. And so some evidence to support this can be taken from this figure from Logan et al., where they had participants complete a memory task, and they had two groups. They had an older and a younger set of adults. And this is an average of some of the experiments they did, but what they were trying to show here is that brain activity in their older relative to younger adults reflected more of a loss of recruitment of these brain areas they were seeing in younger adults. And this little arrow is actually indicating one place where there seemed to be a little bit more of an over-recruitment of activation relative to what they were seeing in young adults, suggesting that there was this non-selective alteration and how brain areas were responding in our scanner to this particular trial event experiment. But at the systems level, we think about things a little bit differently. And so to walk you through what this idea is, so typically folks might try to understand how those brain areas are cooperating across time by defining areas using an a priori atlas like the one you're seeing here, where they can extract a time series of information from each area and then correlate that time series to get a sense of the strength of relationship between 2, 3, or 4 areas with respect to a system that they may be allied to, which we can then get an anchor for how specialized or unspecialized that whole brain organization is based on those system allegiances, right? So here you're seeing a clustering of this blue color representing this visual 2 system from this atlas. And you're seeing that they're more closer together because these edges or these lines here are representing the strength of that connection between brain areas relative to the system. So they're more clustered because they're more strongly related to one another relative to the rest of the brain. But as you might guess from my earlier pitch, as we age, there's a loss of this specialization. There's some evidence from this using a summary measure of organization called system segregation, from David Madden's publication in 2024 showed that while participants at different ages were performing what's called the visual search task, so you're responding to an attentional stimulus in the presence of distractors, that there was a decline in that system segregation measure, suggesting that there was a loss of specialization, at least in terms of the organization of these systems in the brain. Okay, so we had a big picture question then from this, which is this evidence that we're seeing in terms of a loss of functional specialization in brain areas and systems some generalized property of the aging brain that we can find while individuals are performing various cognitive tasks that are tapping different processes? And if so, are they telling us something about behavior? This is a pretty big order, I think. And so what we decided to do was to leverage data from the Human Connectome Project data set, particularly their aging data set from the lifespan sample that they collected, which provided us with 329 participants between the ages of 36 and 100. That's a little young skewed here, that completed 3 separate tasks. First being an inhibitory control go-no-go task. So for those unfamiliar, participants are asked to press a button whenever they see a go stimulus, and then to not press that button when the stimulus is red, so a no-go stimulus. These participants also completed a version of an associated memory face name task, I think from Susan Hookheimer's group, where participants were asked to press a button during encoding blocks whenever they saw a face name pair to indicate that they encode that information. And then following performance on a distractor task that I'm not picturing here, participants were then asked to press a button whenever they remembered the name that they had learned earlier in relation to the face. The last task that they completed was a visual motor response task, where participants were asked to indicate with a button press whether or not the red patch across the checkerboard stimulus was on the left or the right of that checkerboard stimulus. And so what's great about these 3 tasks is that they're tapping ideally or theoretically different cognitive processes, which is a great opportunity for us to ask a question. It also poses a little bit of a challenge because each of these tasks are really designed to answer different questions about how the brain functions to support cognition. And so if we were to try to understand how selective functional areas are and how that change might be occurring with age, we would need some way to, I think, quantify that similarly across tasks. Right, so still thinking about this in the context of areas, not systems yet. We approach this by setting up this idea of canonical activation, which would give us an anchor point, we thought, about where activation should be expected to then understand in our analyses where things might be changing. And so again, there's a little bit of a challenge for this, right? Because tasks like the inhibitory control go-no-go task, we're really looking at the contrast of interest of being the no-go and go stimuli. And what I'm showing here are just Z threshold group maps, where I'm giving you a sense of where peak activation, let's say for like no-go trials, is located in the brain. And so if we want to operationalize canonical, we decided to focus in on those trial types of interest by identifying peak regions responsible or that are responsive to activation for these particular trial types. So that being the absolute value of the top 5% of these maps, which gave us a sense of go and no-go trial activation, recall and encode activation and so forth, which we could then use to ask our first question, which was, hey, if we follow up our analysis of this particular way, want to find some age-related signal. Are we seeing that with this particular approach? So we tested that, and what we found was that this approach, right, using this mask and extracting the average absolute value of activation, all of our participants across all 3 tasks, reflected this idea of age-related decline that we were expecting in these expected areas with increased age, although there's some variability here, right? Which set us up to then ask, okay, so if we know this mask is telling us something about how change in activation in these areas is occurring with age, what about in other areas of the brain not captured by this mask? And so when we asked that question, essentially by looking at the absolute value of activation everywhere else, and we averaged that and we fit a linear model similar to the one before, controlling for 6 years of education as well as canonical activation, we found that at least for 2 of these tasks, that increased age was associated with an increase in activation outside of those expected areas. Okay, so now that we have this measure of activation, expected and unexpected areas, and it seems to be telling us something about differences between participants as a function of age, we felt set up to ask our bigger picture question, still thinking about this in the context of areas of the brain, which we decided to term activation selectivity. The bigger picture question being how deviation is occurring and whether or not we can pull that information from a difference measure between activation in expected and unexpected areas, still using this absolute value approach, because we think that that's going to tell us something about what you're seeing here, right? So for the high activation selectivity individual, this person is most likely showing activation more so in those canonically defined regions relative to non-canonical regions, while acknowledging that for a low activation selectivity individual, one of three things could likely be happening to make that value small, right? That could be a loss of activation in the canonical region, which is maybe what was happening in that go, no-go plots that I was showing you earlier, an increase in activation in this non-canonical region, or a loss of canonical activation, as well as an increase in activation in these non-canonical regions. And so when we tested this measure then to ask once again, is this telling us something about age-related differences? We found that increasing age was associated with a loss of activation selectivity, as that difference in those measures, similarly across all three tasks. But we're still a little bit curious, right, about this variability that you're seeing here. It seems that you can be older and high in activation selectivity, and also be younger and high in activation selectivity. So is this just a reflection of folks showing more canonical activation? Maybe, maybe not. So we decided to run some additional analyses, still thinking about this in the context of the areas, to get a sense of like what kind of spatial variability was present as a function of this measure that we developed, and across these participants in these three distinct tasks. So in order to do that, what we decided to do was to look at the extremes in our data set. So extremes in terms of age and activation selectivity. So we binned 30 subjects into 4 groups, or sorry, and we binned, I can't do math. All right, we have 30 subjects in each of our groups, of which there was 4 for each task. And we looked at the extremes, like I mentioned. In terms of age, this meant that our young adults were 36 to 45 years of age. The older were 60 to the maximum age range that we had available in the sample, which I think was somewhere in the 90s. I think there's one subject that was there. And we took their Z contrast maps. So an example here from the visual motor task, and we vectorized it. So we had areas represented, a series of values, and we just correlated them. So this is one participant, this is another participant, all from the same visual motor data set, to get a sense of, is the fluctuation in the map, RZ contrast map, similar to other participants? You imagine if you're more canonical looking, these maps will probably be more similar. This was the idea anyway. And so when we rearranged it into a large correlation matrix, for each of the bins were present on the X and Y axis, as you'll see, we got this picture. And so our interpretation of this is that if you are high in activation selectivity, you are more similar in your activation profile than you are if you are low in activation selectivity. And what's interesting is that among those that are low in this value that I created, it doesn't seem that there's a similarity in the relationship between participants or activation profile, which means that if you're not high, it's not the case that your deviation from canonical areas is now forming some new pattern across age, which maybe theoretically is what we expected, but it was nice to see this come out in the data. All right, so moving into our next section, now that we've talked about areas, we talked about a measure that we feel is sensitive to age-related differences, and we feel comfortable, I think, at this point that we found something to comment on that in each of our 3 distinct tasks. The next challenge that we had was to understand how to approach this at the systems level. Right, so we've got a measure that we described earlier, we were interested in, and that we think could be useful in quantifying this organization at the whole brain level of different systems in the brain. And so that's what we decided to pursue. Once again, by using a a priori defined brain atlas, means that while individuals performing the task, we get a time series of information for each of these brain areas to be extracted, that we correlate amongst themselves. So we get a large matrix that we can then assign different allegiance to different systems. Once again, to get a sense of this specialization, this is me showing another version of this depiction, where a given system, like say this blue system, and a more modular situation where systems are more strongly connected to themselves relative to other systems. We see this type of sparse relationship that we would imagine with increased age becomes less clear, right? So we see less distinction among systems. And when we tested this in our data, we found that with increased age, there's a loss using our measure here, system segregation of organization among our systems that was similar across all three tasks. This is while accounting for sex and years of education. So now that we have our chief measures, right, that are describing information at the area level and the systems level, we feel like we have a comment now, I think, on what specialization could mean in a similar fashion across these three tasks. But now we want to know some of these bigger picture questions that we were interested in at the beginning, right? Because as I told you earlier, there's been a lot of evidence, including some that I've shown you so far, to say that this loss is affecting areas and maybe how those areas that are interacting, but it's unclear in the same individuals across three distinct tasks if we'll see the same relationship. And so that's what we ended up doing first, what I'm going to show you first. And when we tested that, we found in our first task, the inhibitory control go-no-go task, that increased whole brain system segregation was positively associated with increased activation selectivity. And this is while accounting for age, sex, and years of education. Of course, there's still a bit of variability here. And when we repeated this test and our other two tasks, so that's our associative memory face name task, our visual motor response task, you see a very similar story, and that increased whole brain system segregation is positively associated with activation selectivity. So another way of saying that could be that how segregated or distinct our system's functional organization is positively associated with how canonical their activation profiles were, as we defined in that measure earlier, which I think was kind of cool. This is beyond age, sex, and years of education, just to reemphasize that. And so now that we have this interesting relationship, right, now we want to get to this behavioral piece because, sorry, online, we lost picture completely. I've been touching this. I've never had this happen before. Oh, I could just keep talking now. Believe me, there's 3 plots. Oh, that's smart. Sorry.
Oh, wow.
Okay,
Thank you.
Right. Okay, so now we're going to test and see if these measures that we've been working with are telling us something about behavior in a similar manner across tasks. And so when we did that, the first thing we tested was our activation selectivity measure. Give you a glimpse of the answer here, right? That essentially when we looked at whether increased activation selectivity or that more canonical pattern of activation is related to, in this case, go trial response times, we did not see a significant relationship there. When we repeated this analysis using other behavioral response time measures that we had available to us in our other 2 tasks, we saw a very similar story. That activation selectivity in terms of the way we operationalized it was just not associated with these behavioral response time measures in our other 2 tasks. But we also have our measure of system level organization that we were interested in, that we know is sensitive to age and is related to this measure of activation selectivity they came up with. So maybe that's going to tell us something about behavior. And so we tested it using a similar model. And what we found was that there was no relationship either at this higher, large whole brain level of organization with go trial reaction time or associative memory face name reaction time during recall blocks and our visual motor response time. So, okay. Now, the way we've been approaching this in terms of our systems level measurement is really an assessment of the whole brain, right? And I don't know if everyone, including myself, believes that there should be a really strong signal at the whole brain level for these really specific behaviors in the scanner. So we decided to look at this in a little bit of a more granular fashion. So we recalculated system segregation, focusing in on those sensory motor systems, because we'd seen some other reports that maybe this might be associated with these in-scanner behaviors. We thought, why don't we check that out in our data? And when we did, we found that increasing sensory motor system segregation significantly was related to faster reaction times in our inhibitory control go-no-go task and our visual motor response task, with this trend happening in the space name test. So yay, we have some behavioral association with one of our measures here, which was exciting to us and encouraging in terms of thinking about this product or project and where we were hoping to go. And to maybe in terms of how to think about what activation selectivity can do the way we've been defining it so far. And so in summary, oh, yeah.
How about the accuracy data, not reaction time?
We've looked at the accuracy data for each of these 3 tasks, and I think there's a trend in this task, but not for the other 2. But there looks like there might be something with the out of scanner accuracy measure here. And we're still like chewing over that. These tasks are interesting to work with. So that's a good question. And yeah, I think an obvious one to ask. Okay, conclusions. All right, so big picture, when we started out in this project, I told you that we were interested in this idea of functional specialization and the way people had reporting a loss and alterations with age at the level of area activations, right? And the system level organization of those area level activations. And so we investigated this in these 3 tasks because we wanted to see if there was a common pattern across using some similar form of measurement that told us a similar story to what others had seen in the same subjects. And this is what we appeared to have found with the analysis approach that we pursued to then understand whether or not that told us something about common behaviors that you could pull from these tasks. And when we did, we found a really cool association that I will repeat all of this for you, was essentially that when we looked at activation selectivity as an index of the loss of functional specialization, we found that there was an association with increased age, so it was age sensitive, but it was also telling us a lot about how canonical your activation pattern was in general, right? Based on the definition that we created from those peak maps. And we also saw separately that we were able to describe this loss of functional specialization, the systems level, similarly across all 3 tasks, and that these 2 measures were related. And yet, as I mentioned earlier, right, that really only our sensory motor system segregation measure appeared to, at least in these analyses, be related to behavior. And so with that, I want to thank everybody for your time and the technical difficulties. I think I'm a little early, so I can take other questions as well that folks might have.
So this is a kind of high level question. It's not really directed at you specifically.
Okay.
But more generally, this notion of an area, which is key to your talk, is topographically defined. So for the defining areas, as I understand it, it's a patch of cortex, a parcel of cortex that is in some way different from some other parcel of cortex, based on some macroscopic principle. The issue with that is that areas defined in that way are not functionally homogeneous at all. So you gave the example of visual cortex, for example. Within primary visual cortex, you have a great deal of functional differentiation, depending on, for example, whether you're in a cyto-oxidase rich or area of the cortex, so-called north. So there's a distinction within primary visual cortex between wavelength sensitive cells, cells that are responding in some way to edges or orientation or whatever, as well as cells that have different ocular, represent different eyes.
Okay.
Another case would be like entorhinal cortex. Grid cells are distributed among non-grid cells.
Yeah.
So, and this turns out, at least according to people like Earl Miller, to be a general property of all cortical regions. But neurons within those regions that are macroscopically defined are not functionally homogeneous.
Okay.
In other words, more than one computation is being carried out by these regions that are being defined as areas at a macroscopic area.
Yes. So just to make sure I'm anchoring, so you're giving cellular examples and you're talking about when they're functioning together as a unit.
Yeah, what I'm saying is
individual cells and their neighbor cells aren't necessarily doing the same.
Exactly right. So,
which I agree.
So
Yeah.
What is the point then of using area as a unit of analysis when within that area, you've got functionally heterogeneous computations being carried out.
Yeah, so I think that the question you're getting at has to do with how I'm thinking about it in the context of what I can see with functional magnetic resonance imaging, and
Sure.
how I'm thinking about distributed processes emerging from these contrast-based analyses that we do, which is really the limitation, I think, to what I can mean by areas and what I can mean by this change in specialization with age. And that's about where I'm sitting.
I understand that, but what
Yeah.
I'm saying is you can't, the fact that you have a tool that doesn't do the job that you need it to do is not an excuse for using something like area as a fundamental concept when it probably isn't. I agree, fMRI and structural MRI don't
Yeah.
allow us to characterize these regions at the level that we need to characterize them.
Okay, so
But
yes, I mean, I agree with everything you're saying. I'm not sure, so I don't want to confuse anybody to think that I thought that like some patch of brain only does one thing, like I'm localizing function, for example. It's not what I'm defining. I don't think that's what you're saying either, but I just want to be clear. I totally agree with you, and I think that in terms of my hierarchy example, that's kind of what I meant by that, right? That we can ask these questions in different ways at different levels. And then if we were at this cell level and I had access to that in humans, probably be talking about this in the exact way you're talking about it. They don't have access. I have access to essentially this type of information with the functional magnetic resonance imaging tool that I have. And I believe that there's something meaningful about how we see activation patterns emerge in individuals. That change is indicating something to us about how areas are maybe responding with age differently than they were before. And that's really the extent to which I can discuss that. It's not as specific as I would like it to be. All I can really do is just comment based on how consistent a particular pattern appears to present while individuals repeat the same behavior. Right. So that was a long-winded explanation. But did that answer or give you a sense of?
yeah, we can talk about it later.
Yeah, yeah,
And
yeah.
I think it partially answers it, but not completely.
Okay. Maybe people have comments or add to that? No.
So I wonder, kind of more on a practical level, but you had the 5% that are most selective for a task,
Yes.
right?
Yeah.
And then was that defined for the group? And
We did it a
that
couple of ways.
Because if it's not, if there will be a lot of variability there, and in a way, those aren't, say, areas, right? Those just happen to be the most related to the task.
Yeah, I think that's
And they
yeah.
could be different across different individuals or systematically different across age group.
Right. Yeah. Which might be explaining some of the variability we see in the data as it is, right? I don't know if we had a really small sample, if it would be as clear, at least some of the associations we're showing here, because there's so much variability of this approach that might be attributed, I think, to what you're picking up on. Yeah, I agree. Other? Okay.
Are there any questions online?
No questions online.
Okay, great, thank you. Just a second. Happy to wait.
Keep your feet well to the left.
Yeah.
I know. All right, we will have our first data blitz. We're going to have Francisco Sierra, who is from the PSOC Lifespan Neuroscience and Cognition.
Hello, everyone. So my name is Francisco Sierra, and I'm going to be talking about my research. This is part of my dissertation. So it's titled Testing the Theory of Working Memory Adaptability on Different Language Groups. I want to see whether this theory generalizes in different language groups, such as monolinguals, bilinguals, and polylingual. So first, I'm going to walk you over what is the theory of working memory adaptability. So this idea is that we have a focus of attention in working memory, and then we have a sort of like a passive outer store where we hold some items that we later use or we later can access to continue working with them. And there's this idea also that with practice, you could increase this focus of attention, but it also depends on what kind of condition you're practicing and testing. For example, if you're practicing and testing with a predictable condition, then you can expand your focus of attention to maybe 3 or 4 items. So there's this old idea that working memory has 7 plus or minus 2. Well, that has changed with recent research, and now it says that the current research says that it's up to like 3 or 4 items. But that's only if it's a predictable condition. And what do I mean by predictable? It's that you can basically tell whether an item or a kind of item is going to follow in a specific sequence. But if the task is unpredictable, the focus of attention only remains confined to one item. So it depends on what kind of task you're doing. And this also depends on your set size or difficulty level. So depending on how many items you have to keep track of or how many elements, that could affect your focus of attention. So if you go to a larger set size, as you can see there, present, the response time will be increasing, but it's going to increase more if it's unpredictable condition as opposed to a predictable one. So now talking about my study, and you need to be fast. So I collected data from 61 participants, but I had to delete one participant because they provided incomplete data. So participants were mostly young adults, mean age 20, and I had 21 monolinguals, 28 bilinguals, 6 of which were fluent in Spanish, and 11 polylinguals. And the kind of stimuli that I was looking at with my working memory updating task, which was my variable of interest, were cognitive plus plus words, cognitive minus minus, and uncognitive words. But also we had digits, cars, shapes, and birds. And this working memory updating task, essentially, they have to keep track of different items and different locations. And they have to remember not only what item they saw, but also keep track of all the different locations and their asset outer storage. So just to go over the results very quickly, there was no significant main effect on language group for cognitive efficiency. And just really quick, this measure of cognitive efficiency is the difference between the Z-score of accuracy and the Z-score of reaction time. So it gives a accuracy speed trade-off measure, and it combines both accuracy and reaction time. But there was a significant interaction between language group and stimulus type. So essentially, I found that bilinguals had a significantly higher cognitive efficiency for the cognitive words, for the plus plus and the minus minus. So cognitive words, really quickly, these are words that share orthographic and phonological similarity to words in another language. In this case, the reference language was Spanish. And cognitive plus plus, as you might or may not imagine, these are words that share a lot of phonological and orthographic similarity. And the cognitive minus minus words, not so much, but they still are cognitive words. And our findings are basically that these theory of working memory adaptability is generalizable to different language groups. And you can see with this biolink plot how there is a clear decrease from 0, 2, and 3 difficulty levels for all the types of stimuli for the different language groups as well. Do we have any questions?
What's the difference between cognate plus plus and cognate minus minus?
Yes, so cognate plus plus, these are words that share a high degree of phonological and orthographic similarity. In other words, the words pretty much are written the same and sound almost the same to the words in the reference language. An example of the cognate minus minus words, they share less similarity, but you can still recognize the word from the reference word. Whereas non-cognitive words, those are words that still mean the same thing, but they're written and sound completely different. Yes. Any other questions?
Actually, we are going to have to move on to the next one. So have a round of applause. So we have time for one question after each presentation. And next up, we have Umi Coates from the Wig Neuroimaging Lab.
There's a laser pointer there. You wanted to use it.
Hi everyone, Mooney, and today I'm going to be talking about my project on the role of life course environmental exposures on midlife white matter brain health. So your environment is made up of a bunch of different factors. It's your social circle, it's your diet, it's where you work, and it's where you live. Exposal models of health argues that it's a culmination of these different environmental exposures that interact with your genome, leading to differences in health outcomes, like Alzheimer's disease, for example. These exposures can also vary and accumulate across one's lifetime, which is why it's important to look at the effects of timing on these exposures, hopefully with the goal of modifying or changing brain aging trajectory. So there are two different types of challenges that can come with looking at brain aging in this way. Firstly, traditionally, education group has been used as an indicator of environmental exposures, but education group is also a pretty broad marker of some of these exposure variables. And it's also that midlife, when we look at midlife and we're studying aging, it's difficult to look at how exposures vary across the lifespan. So my project will talk a little bit more about how I can overcome some of these challenges when looking at environmental exposures and how they vary. So in this study, we're interested in white matter hyperintensities, and these are lesions that accumulate in the white matter with increasing age. They're also associated with Alzheimer's disease risk and cognitive decline. And so in my project, I'm interested in how environmental exposures relate to adult midlife white matter health and its change across time. And if we can identify a particular life course period that is important for predicting midlife white matter health. Firstly, because we're interested in the effects of environmental exposures and timing, we wanted to compare two different time periods, childhood and present day. So for childhood, we looked at parental education as our marker of childhood environment. And then we also looked at current education as our marker of present day. The dark colors indicate college degree and above, and then the lighter colors indicate less than a college degree. And then we have white matter hyperintensities on the y-axis. What's really cool is that we found that highest parental education is strongly predictive of midlife white matter health, meaning that in our sample, for middle-aged adults, if you have a parent that did not have a college education, you're more likely to have more white matter damage than a peer whose parents did go to college. And surprisingly, we found that this effect was not found in current education. So it seems to be a very strong effect of childhood environment in predicting later life white matter trajectories. And all of these analyses were controlled for current education as well. So again, the more traditional approach is to look at education group, but it's very broad, and we wanted something that's a little bit more granular in understanding these different types of environmental exposures, which is why we created something called the historic neighborhood deprivation construct, or the HNDC. So the historic neighborhood deprivation construct is kind of similar to the area deprivation index if you're familiar with it, but essentially we collected 11 standardized census tract level variables going back from 1970 to 2010. And we collected variables that we know are related to neighborhood quality. So things like percent unemployed, percent with a high school diploma, and the median household income of an area. We collected all these variables and then we created a score, and then we mapped that score to each participant's recalled life course address. And then from that, we averaged those scores across different life course periods. So we looked at childhood, we looked at adolescence, and then later we broke down adolescence into early and late adolescence. We also looked at young adulthood and middle adulthood. So what did we find? So we found that childhood and neighborhood quality was strongly predictive of midlife white matter health. And again, this is also while controlling for current education. So being born in a highly deprived area is strongly predictive of more white matter damage when you are a middle-aged adult. We found that when comparing the effects of these different life course periods on white matter health, so again, when taking into consideration where you lived as a teenager, where you lived in college and midlife, where you live now, childhood is still the most predictive of white matter hyperintensities. So what does this all mean? So in conclusion, we found that early life, particularly childhood, might serve as a critical period towards predicting midlife brain health. And we didn't show these analyses here, but we also did some repeated measures longitudinal analyses, and we found that childhood environment was only predictive of intercept differences, but not slope differences. So childhood seems to affect where you start, but not how you change across time. And in the future, we're interested in looking at this association with modifiable resilience factors, so things like lifestyle, stress, physical health, inflammatory biomarkers, to see if potentially we could, if people who are born in adverse childhood environments might benefit from having a better lifestyle and better health against some of these effects on white matter lesions. Thank you.
Curious, how were you quantifying white matter health in those early figures?
So we're using white matter hyperintensities. So we quantify that with T1 images. And so these are lesions.
Oh, that's okay. Yeah, here we go. That makes sense now. Thank you.
Thank you. All right, next up we have Dr. Gelson Wong from the CVL Aging Well Lab. That's
So for the work I'm presenting today is actually going to be a deep dive into some of the work I've actually presented here at CVL not too long ago, whereby we are looking at how decisions under ambiguity are affected based on the different types of decision costs. But specifically for this presentation, I'm actually doing a deeper dive onto one chunk of it, which is how decisions under ambiguity can be, how it affects cognitive effort discounting. So specifically this, even though ambiguity is something that's very ubiquitous in a lot of the decisions that we do, the research in this area has been very, very scarce. And so by extension, because of that, we don't have a lot of computational models that tries to understand the mechanisms behind how people are processing ambiguity and effort expenditure when they are making their choices. So really, this is my attempt at trying to actually try and identify a model that is able to best describe how we could actually, how people might actually process ambiguous effort expenditure costs when they're actually making choices. So if I just direct you to the figures on the left here, so the test is pretty simple in terms of we're using a two-task paradigm. So we ask participants to choose either a smaller reward for a smaller effort option, or would you rather invest more effort and then you can get more money back in return on the right hand side here. And ambiguity is introduced here by masking the exact cost of the actual level of the effort that you're required to receive the amount of money. And what we have in terms of just looking at the behavioral data here, there are two things to note. One is that the degrees of ambiguity does seem to show an effect on a participant's rate of accepting the larger option across the different levels of the different difficulty effort levels of the task. And so what happens is that we see people are showing more aversion to ambiguity. The second thing to note here is that this is a bit of a differential effect. So we see that more pronounced when the task is, when the option is harder, but when the option at effort level is easier, that we don't see much of an ambiguity effect. And prior to that, what I did here was I tried to use a hyperbolic discounting function, at least a modified version of it, to try and see whether or not I could get at these ambiguity preference estimates. Now, one thing though here is that there are, in terms of effort discounting, there are other models that studies have found that they are actually much better at measuring and estimating an individual's choice preferences, especially here if we look at the, so for example, one is a quadratic model. Here I have an example, it's a sigmoidal function here. And just looking at the two graphs here, that's trying to map out how subjective value changes as the effort level increases. You can see the way that it's going to actually compute a subjective level for the same effort is going to be different, and that's going to affect the way that people are probably going to be making choices. So why is this important, right? Well, two main things here is that, first of all, if you're using a misidentified model or a poor fitting model to try and measure and estimate these preferences, you're going to get parameters that are going to be biased, or they may not be actually really good at estimating the effect that you're looking at. Because when you're using a poorly fitted model, a lot of the structural model is going to be affected by how the parameters are trying to compensate for this poor fit. Another thing is we're trying to model behavior and trying to predict behavior. If you're using a poorly fitted model, you're not going to be able to predict future behaviors as well. And so
for that, what I'm trying to do here is I try to look at how the numbers, the percentages numbers just got weird here. Never mind. So what I did here, using the two models that you saw, the hyperbolic additive model and the sigmoidal model that I just presented before, I also include another two more models here, which on the right here are threshold models which are variations on the one on the left. So the reason why I accounted for these threshold models is because, as I've shown in the data before, the effects of ambiguity seems to only present itself at different effort levels. And what I'm trying to do here with the threshold here, so I'm not sure if you can see my cursor here. So I have a threshold X here, which means that only when I reach a certain level, effort level, then do I apply a computation that considers ambiguity. And so my percentages here are messed up, but this is basically the ratio distribution of the best fitting models based on after using a maximum likelihood estimation approach, as well as using an AIC as the criteria for best model fit. And so what I do find here is that a lot of the participants are actually best fitted by a sigmoidal function, whether or not is it an additive function with a threshold or without its threshold. And so another thing to note here is that also the fact that a lot of them are actually being best identified by the threshold model means that there is also a way that this means that it is important for us to actually account for the differential impacts of ambiguity effects across specific levels of the task that we're presenting with them. And moving forward, what do I have planned for this is perhaps to revisit some of the analysis that I've done. So perhaps now that I'm using a specified model, I might actually be able to see different effects that I probably would not have been able to see using a hyperbolic function model. At the same time, there are now different parameters in these new models that I wasn't accounting for. And perhaps I could also look at how I could try and maybe hypothesize how these could be affected by different variables in the set. And that's it. Thank you.
Okay. Next up, we have Rory Massett from the Wig Neuroimaging Lab.
Okay, so yeah, I'm Roy. I'm a PhD student in the Wig Lab, and today I'm going to be talking about this project that I've been working on called Spatiotemporal Heterogeneity of Alzheimer's Disease Related to Functional Brain Network Reorganization. So as many of you know, our lab is interested in how brain networks change across age. So our lab has shown and repeatedly shown that brain network organization changes with age, specifically this measure of system segregation is reduced with age. And more recently, we've also shown that on top of the effect of age, there's also an effect of increasing dementia severity that further is associated with reduced system segregation or reduced network integrity. You can think about it. The thing about when you're considering disease like Alzheimer's disease is that it's a disease that evolves over the course of years or decades. So when you're measuring someone, you're capturing them at some point in the disease course. What has also been shown, mainly through structural and pathological measures, is that there are several different subtypes of Alzheimer's, biological subtypes of Alzheimer's disease that have different sort of progression patterns. And so these are both subtype and stage are important things to consider when sort of quantifying differences between disease individuals and healthy individuals. Less the question of how functional network organization varies in terms of time and space across the disease course. So to get this question, we got 345 amyloid positive subjects from the Alzheimer's Disease Neuroimaging Initiative. And for each of these subjects, before we were looking at whole brain organization. Instead, here we were looking at the organization or the segregation of individual systems. And then we use this tool called subtype and stage inference. And I don't have time to talk about it, but the TLDR is that it predicts the subtype and stage of each subject that you're giving it, according to the features that you're giving it in the case system-wide segregations. So from this data-driven method, we derived two subtypes. So you can appreciate in early stages, subtype 1 is sort of what you expect when you think about Alzheimer's disease pathology. Early on, it affects association systems, particularly the default mode network. So you can see that those systems are being affected here. Here, these are showing which systems are affected by this stage. Redder colors are lower or quote unquote worse network integrity system segregation. By contrast, in subtype 2 early on, we actually see sensory motor systems being affected, which is the opposite of what we usually think about with typical Alzheimer's disease. Typically, we think about these sensory motor regions being relatively spared until very late stages of the disease. So if you play this out, you can, by the end, the algorithm assumes that all regions are affected by the latest stage of the disease. But even there, you can see that compared to the two, when comparing the two subtypes, again, these association regions, default mode, etc., are relatively spared in subtype 2 in terms of their network integrity. And if you collapse across stages, just to show which systems have higher network integrity in each subtype, sorry, lower network integrity in each subtype. Here in subtype 1, it's roughly following this association versus sensory motor system distinction, where association systems are more affected in subtype 1, but the opposite case is in subtype 2. So just to recap, we can group Alzheimer's disease subjects into these two subtypes based on resting state brain network organization, and they're generally following the sensory motor and association system distinction. And we think that reorganization of these distinct sets of systems may be a link between the sort of clinical heterogeneity and biological subtypes that we see. And so in the future, we're planning on linking these subtypes explicitly to those clinical variants and these structural and pathology-based subtypes. That is the usual approach. So yeah.
So I may have missed it, but those most of the participants in the database would fall into 1 or 2, or most of them would actually not be classified cleanly as 1 or 2 subtype.
Sure. So it is probabilistic. So each subject has some probability of belonging to each subtype and each stage within that subtype. So the way it works is that it takes the subtype that is most likely, and then within that, it finds the stage that's most likely. But all the probabilities are above chance. There's sort of this characteristic shape of the probabilities where subjects at really early and really late stages are more ambiguous. So there's sort of this inverted U shape where the subtypes are more distinct in sort of the middle.
But it kind of forces them into 1 or the other.
Yeah, that's right.
Kind of stage.
It doesn't. There are some subjects it will assign to neither if they don't fit into either sequence. Yes.
Thank you. And last but not least, we have Parker Young from the Offen Lab for Cognitive and Brain Development. Oh, sorry, I'm good with that. Yeah. Okay, perfect.
So hello, my name is Marco Younger, and I'm going to be explaining a preliminary project that the Open Lab has been working on. So it is known that adults tend to review more material more effectively than children and adolescents, but the neural basis for underlying this advantage remains unclear. Maybe it's because as we move through the developmental timeline, we find new ways to organize information that is presented to us. So one contributing factor may be the age-related refinement of mnemonic strategies, such as chunking. And chunking refers to the reorganization of individual items into more meaningful and larger groups. So because chunking is so effective for verbal learning, we presented 105 participants with the 5 free recall trials from the California Verbal Learning Test children's version. So these trials are repeated throughout the test, and participants aged 5 to 25 were verbally presented this 15-item shopping list, which comprises of 3 semantic categories, and then participants were asked to pre-recall their responses on what they remember from what was verbally presented. We quantified a string of recalled words as a chunk if they were organized as a semantic category or a semantic chunk, which would be 2 or more words recalled from the same semantic category. And then we quantified serial chunks as words that were remembered consecutively from the original presented order. So as you can see, we have ball and chalk. This would be a semantic chunk. And then we have, for example, like berry and goggles, because they're represented in the same order. In this case, 4 words. We can see them quantified as a serial chunk. So then the typical trend for the CVLT is to see an exponential increase in the overall performance across trials as the lists are repeated over time. So here we can see that the number of chunks formed was similar across children, adolescents, and adults. However, we can see for adults that the number of chunks in trial 4 and trial 5 decreases over time. So because of this, we were really interested in the more novel approach of this study, which is to see the content inside of the chunks formed. So we quantified chunk fragments as prior components of prior chunks that were brought over to the trial that we were attempting to analyze. So as you can see, that ball and chalk that I was explaining earlier, it's a component that's being brought over from trial 1, and thus we quantify it as a chunk fragment from trial 1, that first chunk. Okay, and because the number of chunks for all the age groups stay relatively stable, and the number of chunk fragments increased, as we can see in the second plot, we can see for adults that as the trials progressed, the number of chunk fragments increased for them. We were trying to understand why there is a robust difference in this trial 1 versus trial 5 comparison. So for further analysis, we focused on this robust difference. And so then we plotted based on the continual variable of age. And this is just to show you again that there is a significant correlation between the mean number of chunk fragments from trial 1 to 5 and that age category. So it just kind of helps us to understand this this plot before. And so when it's plotted, we can see that significant positive correlation. And so now, since we were interested in understanding the neural basis of chunking, we decided to build off of prior research from our lab that shows the link between prefrontal cortex maturation with the use of mnemonic strategies, specifically the spontaneous use of these strategies. So the one region that is shown to link with the spontaneous change of mnemonic strategies is the the rostral-medial-frontal gyrus. And so what we did is we looked at cortical thickness of these regions using structural MRI images. And cortical thickness of this region was found to be negatively correlated with the mean number of chunk fragments. And so what this ultimately helps us to understand is that this might be in part to the maturation of the prefrontal cortex, which we know throughout development shows cortical thinning. And then taken together, we understand that these findings suggest adults utilize and they are able to effectively use prior components, and they're able to use the effective mnemonic strategy of chunking to encode information into fewer but larger chunks. And we also find that PFC cortical thinning is linked to effective online processing of verbal information. And so future research on this is we're trying to look at how the age-related cortical thinning of these regions in the prefrontal cortex leads to an understanding into why adults may have a developed skill in online processing of it.
In the analysis of frontal thickness and chunk fragment, did you control for age?
So we did not control for age.
For age?
For age. Okay, sorry. So I apologize. Yes, we did control for age,
So
but I'm not 100%. Like, yeah.
that would be necessary if you
Yeah.
want to show that there's an independent relationship between thickness and chunking.
Yeah.
Okay, thank you.
Want to-
But that's it. Okay. All right. Thank you.
Thank you.
Thank you. Right. Thank you, everyone that presented today, and thank you all for attending.
Thank you.
Thank you.